So welcome to the Science Education Corner. So as you know, the Science Education Corner is to gain some knowledge about science in our daily life or in some maybe modern technologies that are available right now. So as usual, the person who will help you with the session of the Science Education Corner is your brother. Who? Me. Yeah. So you can see my picture over there. And yeah, you remember my name, right? So the topic today is microarray for our science corner. So actually I want to do this topic because of the question you asked me, that your curiosity of the question you asked me the other day on what was I doing at that time, and you wanted to know a bit on microarray. So microarray is actually a quite uh, high knowledge topic, but because of your curiosity, so I try to simplify it for you so that you can understand more better on microarray. So what is microarray? Sarah, what do you think? What is microarray? So before you want to discuss microarray, there are two terms that we need to know on which is linked to microarray. Both are EST and cDNA library. So I know these terms are a bit new for you. So EST is an express sequence tag. Do you remember last week, Sarah, that I taught you about the DNA sequence of ATCG? Okay, so ATCG is a sequence of the DNA, which is a gene. So express sequence tags is a sequence that is expressed in a gene that are tagged by that are tagged to identify it. So let's say that maybe I want to find a gene to study why my hair is white. So during the at the white hair, I will use the tag to label which gene is expressed when my hair is white. So you get it. So where do these ESTs come from? Express sequence tag. So they come from cDNA library. What is a cDNA library? Well, to make it easy for you, library. Okay, have you been to a library, Sarah? I know you have, right? Because I asked you to follow me the other day to the National Library. So as you know, library is has many books, right? Has many books, has many information. So that's the same as cDNA library. So cDNA library is a collection or a storage of complementary DNA, cDNA. So these complementary DNAs are actually from messenger RNA. So how do I tell you what is messenger RNA is, as I said earlier, how do I find that what grows my hair white? So mRNA are like the messenger, that is like the SMS. So the genes send the SMS to make my hair white, to my hair. So microarray, so back on the topic microarray. So microarray is actually the collection of a microscopic cDNA spots attached to a solid surface. So let's discuss a very important tool in microarray, which is microarray glass plate. What do you think microarray glass plate looks like? Let's see. So you can see that microarray glass plate is square or rectangle, and it has actually this one has actually one 1200 slots in it. So each slot has actually a cDNA in it. So it has each gene. So let's say that the number the genes that make my hair white is this area. So we will study on this area, of course, to analyze later. And this whole 102,000 spot is the sequence of my whole DNA. So actually, in these modern days, many biotech companies already have a template. So let's say that I want to do an experiment on a monkey, or maybe a simpler version of a rat. Make it simple. So biotech companies already have the template. So I just buy these glass plates to make it easier for me to do my experiment. So usually during the analysis, only before the experiment all will appear white, but after the experiment, during the analysis there will be like the colors of red, yellow and green, which I will explain later in the experiment. So red is for I'll explain later in the analysis of experiment, but it is quite important in the microarray class plate. So how do we use microarray? So there are actually 7 steps, which I already minus the ones to create a microarray glass plate, because I have bought one to help us with this experiment. So 7 steps will be needed to do a microarray. So Sarah, what experiment do you want to do today? Do you know what are microarrays used for? So a basic experiment that we can do is white hair versus black hair. So we want to know the genes that express. So the expression of genes is one of the use of microarray. So we want to know the genes in the white hair, what is expressed, and the genes expressed in the black hair. So how do we do these experiments of the microarray? So firstly, we have to take a sample. So yeah, let's take a sample of my white hair over here and my black hair over here. So after that, we have to extract the samples. We have to extract the mRNA of the samples. So the mRNA, why do we extract the mRNA only? Because it specifies in the expression of genes at that time. So it is important us to know what as the genes are expressed. So after I extract, so the hair was in these tubes, so I used the extraction buffer. So usually extraction buffer has a label, but for the purpose of this for to teach you, I used a bottle with no display. So after that, after I extract that, there will only be left with mRNAs. So the next step, the third step, is converting the mRNAs to cDNA. Why do I need to do that? Because naturally mRNAs are less stable than cDNAs. So cDNAs will increase the success of the experiment. So how do we do that actually? We use an enzyme called reverse transcriptase. So Sarah, enzyme, if you're not familiar with that, is actually something that helps us to speed up the experiment. So it's more to like when you're playing a game, you have a cheat code to speed up the game. So that's the same with enzyme. So we use enzyme to speed up the experiment. So the next step is dye. So we will dye these both of the tubes with different colors. So in this experiment, I dyed the gray hair tube on my left with a red dye, and I dyed the black hair tube with a green dye. So after that, we mix both tubes and hybridize it and wash the excess of the microarray. So how do we hybridize it? The mixed tubes will be poured onto the microarray. After that, after they were poured, it will hybridize automatically in the microarray. After that, we will wash the microarray in the washing solution to remove the excess of the cDNA. Okay, so after that is a process called scanning. So scanning, we will scan it using two lasers, which is the red laser and green laser. So it will be done one by one. So after the scanning of one laser, the data will be kept in the computer for further analysis. So now for the analysis part. So this is like the glass plate after the experiment. So to ease you up, I have found an example on the research for the cancers. So let's just applicate this in our experiment. So for the red dot is actually upregulated in gray hair. The green one is actually downregulated in gray hair. The yellow dot is expressed in both gray hair and black hair. The black one is not expressed in both cells. So as we can see in this glass plate, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So there are 15 red dots over here. So these genes, so one dot, as I said, represents one genes. So these genes, Sarah, is why my hair is gray right now. So to create a medicine, or maybe if a person wants to do a hair supplement to reduce the gray hair, they can actually suppress these genes, these red genes, and promote these green genes, these green spots. What are the green spots? Is the ones that are expressed during the black hair. So these genes make the black hair grow. So the yellow genes are expressed in both cells. So these are actually like the constant variable in what you learn in your school experiments. So the black ones, in the other hand, are the ones that are not expressed in these genes. So maybe that these genes are actually maybe expressed in other situations or other cells or other environments, because all the genes of the genome are there, and not all the genes will be expressed in one environment. So that's all from my the science corner from your brother today. So thank you for watching, Sarah, this video, and I hope you gained some knowledge over there. So any questions you can ask me later. Thank you.